Hello everyone, welcome to my channel. Today we will start the Papers of the Week series, episode 3. I will talk about one paper. It's the genome-wide CRISPR screens with single-cell transcriptome readout. It was published on Cell and a very important paper in CRISPR field, and it pushed CRISPR screens to single cell level at a genome-wide scale. Okay, and so before we start, I would like to introduce why a single cell CRISPR screen is necessary, and what's the advantage over the regular CRISPR screens. Okay, so regular pooled CRISPR screen is we infect the cells with multiple sRNAs and targeting different genes, okay, and then pour them together to perform the selection, and the readout is is sRNA counts, okay. If the count of the sRNAs growing, how to say, increase compared to the control, then that means knockout of these genes will cause the cell growing faster. While if the sRNA counts decrease compared to the control, that means this gene knockout will cause a cell death. Okay, there will affect the cell growth. So this kind of readout will mainly get those genes that affect the cell growth, or if we use drugs, then we can get the genes that cause a cell resistant drug or sensitive to drugs. So this kind of readout is kind of limited because there are many gene knockout will have no effect, no such effect on the cell viability, but it will affect other cell phenotypes, such as development and other gene, other phenotypes that are cannot be measured through the cell growth. Okay, so in this way, we can use a single cell CRISPR screen, the same experiment settings by lentiviral guide RNA infected cells and pour them together, and then we do this single cell approach, and each cell we can measure its transcriptome, and by comparing to the controls, either with non-targeting sRNA or the AAVS sRNA, we can see how the gene expression changes, downstream gene expression changes. And then at the same time, we can measure the sRNA sequence in the transcriptome. So we can link the gRNA sequence to the transcriptome readout. Then we can get a very informative readout, right? We can see how the knockout of certain gene will affect the downstream gene expression change. Okay, and this is an advantage over the viability CRISPR screens. Okay, so then I think the major the key point for the CRISPR single cell CRISPR screen is how to link the sRNA sequence and the transcriptome, the single cell RNA-seq result. Okay, then there are two designs for this kind of experiment. One is called Perturb-seq, and it was published on Nature Method 2017, and they modify this regularly used lenti-guide pro vector for the CRISPR screens by integrating human U6 promoter and sRNA sequence. This is called the gRNA cassette into this SINE 3' LTR. So with this setting, the the gRNA cassette were copied during the lentiviral integration. Okay, one is this copy as a 5'. This copy will express the sgRNA sequence, and then it will perform the gene knockout or gene knockout together with the Cas9 protein. Okay, so this one. Another one is at 5', and it will express together with this transcript at this EF1A promoter, and this sequence will captured in single-cell RNA-seq. Okay, so this is important. So you can directly capture this sgRNA sequence, and you know this sgRNA will cause certain gene knockout, and then you can also get the single-cell RNA-seq of all the other genes, the transcriptome, okay, and then you can link the sgRNA sequence to the single-cell RNA-seq data. Okay, so this is a very clever design, and another one is called Perturb-seq, and Perturb-seq is also proposed earlier at 2017, but at that time, this the design is not very it's not optimized, and it cannot directly capture the sgRNA sequence. So in later 2020 Nature Biotech paper, they further optimize this system, and allowing the direct capture of sgRNA sequence and single-cell RNA-seq. What they do is introduce capture sequence into this sgRNA sequence. No, not the sgRNA sequence. It's a constant region of the sgRNA sequence. Okay, so actually the sgRNA sequence is consists of the sgRNA target sequence and this consistent region. Okay, so by insert the capture sequence at this region, they can okay, so this is the capture sequence, right? And which is a reverse complement to the template. Okay, which with the reverse transcript, with a reverse transcription, they can capture this sgRNA sequence together with this sgRNA sequence. Okay, then at the same time, they will also keep the UMI and the cell barcode. So the UMI and the cell barcode is to identify which cell is this the identity of the cell. Okay, so with each cell they have a unique cell barcode, and with each transcript they will have a unique molecular identifier. Okay, so with this approach, they can link the cell barcode and the sgRNA sequence. Then they can link the sgRNA sequence, the gene knockout, and the downstream sgRNA single-cell RNA-seq data. Okay, so this is another way to capture the CRISPR screens, to capture the to measure the single-cell RNA-seq and capture the sgRNA sequence at the same time. Okay. Oh, then come back to this genome-wide single-cell CRISPR screens. They use this capture this Perturb-seq and with direct capture of the sgRNA sequence as experiment for their experimental design, and they perform this genome-wide screen at with three settings. One is 2 in K562 cell line, one is RPE1 cell line. And for this one, the target genome-wide, targeting all the expressed genes. So in total is over nearly 10,000 genes that are expressed, okay? And this requires like more than 2.5 billion cells, okay? So this that's a lot of money. They are very rich. And with these settings, they can get this kind. So this is the main readout for this screen. You can treat it. You can see it is a very big matrix. So with index is the gene expression. So this is the single cell transcriptome. Okay, for the expression of all the genes in the genome. And the columns are the cells with linked perturbation. So each cell will assign the one sgRNA, right? So all the cells have for each cell you have a sgRNA, and you can link it to specific gene perturbation. And with this matrix, what they can do is to cluster the perturbation to define the gene function, right? So they can measure the correlation between each gene perturbation pairs by comparing their gene expression profile, and then each pair. So this is a symmetric matrix, right? Each symmetric matrix. Okay, so for those genes with a similar function, which we are called a similar downstream gene expression profile, they will the distance, their correlation is very high, so they are similar to each other. They will form clusters, and at the same time, they can do clustering at both the perturbation and the gene expression. So in that way, they can link the perturbation to the downstream gene expression change, which pathway was by perturbating which gene set cause the gene expression change of this gene set. Okay, so in this way they can so this is they can also say this is called genotype and phenotype mapping. Okay, and also with this approach, they can measure the RNA splicing and total RNA content, transposon elements, and chromosome instability and cell cycle. So these can all be measured in single cell RNA-seq data, and with this we can know the drivers, because you can link the sgRNA, the gene perturbation, to these composite phenotypes, right? Then you can know which gene, the knockout of which gene will cause the changes of these phenotypes. Then you can know the drivers of these important phenotypes. And first, they measure the global efficacy of the CRISPR knockdown, and you can see the fraction of mRNA remaining. Majority, I think, 80% of the more than 80% of the sgRNAs will cause over 80% of the gene knockdown. Okay, so that means the efficiency of this sRNA library is good. And to deter, they use the Anderson-Darling test to detect the individual differentially expressed genes. That means a knockdown of this gene will cause significant changes of these genes in cells. And the number of the differential expressed genes are highly correlated with the growth phenotype. This is reasonable because the more the more gene exchanges, no, the more gene expression change the perturbation cause, then the higher probability that this gene knockout will affect the cell growth. Okay. So while this is true, but there are still 771 genetic perturbations with almost no growth phenotype, but significantly transcriptional phenotypes will cause. So that means this 700 more gene perturbation will cause gene expression changes that are not very closely related to the cell growth, maybe to the cell development, etc. Right? So this is also the advantage of the knockout, the single cell CRISPR screen compared to the cell viability screens, right? And then with this matrix, so they have like 1973 perturbations that has strong phenotypes, which will cause a significant downstream transcriptional change. Okay, and also in the column they have 2,319 highly variable genes. So these genes are significant changes upon the knockdown of these genes. And with this, they can do the perturbation wise, pairwise perturbation clustering, as I mentioned previously. So with this, you can find the pathways or protein complexes that are working closely to each other. They will cause a similar downstream gene expression change, right? So so you can see here the gene pairs that in a quorum complex. So this is a database of protein complexes. The protein complexes have much higher correlations, gene pairs, compared to all the gene pairs. So this is reasonable, right? And this is also showing in the string score. Strings are not database for gene and gene-gene interaction. And with a higher string score, that means it's higher. These two genes are highly predicted to have interactions with each other. Then they will have a higher correlation in this map. So this is also reasonable. And another approach, another advantage of this kind of analysis is we can find those poorly annotated genes, which are in previous annotation, maybe it's not well annotated, but if we find one genes that are highly correlated with other gene sets, that means they will have a similar function, right? So in this study, they find these 10 genes that are correlated with ribosome RNA synthesis. Okay, this this 1, 2, 3, 4, 5. So these 5 genes are related to 60S ribosome RNA, and this is 40S RNA. So these 4, and they find this all these genes are not poorly annotated in the database, and with experimental validation, they find 9 out of 10 genes knockdown will cause RNA rRNA synthesize a different change. Okay, 28S divided by 18S. So this knockdown will cause this over the 28S increase, while this will cause 18S increase. Okay, there's only one gene. This gene will cause no significant change compared to the control. Okay, so this is just another application of this approach. And another one is to identify a novel component in complex. Okay, so in this study, they find this integral complex. Actually, they find not all the components have a similar effect. They have 3 major clusters, and these 3 major clusters are corresponding to different functional modules in this complex. Okay, this is very convincing result. That means in this strategy can find those functional modules in the same complex that corresponding to different function, right? And actually, they find a new component in this module called C7ORF26. Okay, module. This gene, and they have very similar expression profile to INTS 10, 17, and 14. Okay, 13 and 14. Okay, and by doing experimental validation, they find this gene knockout will greatly affect the expression of INTS 10. So that means it may form complex with this gene. Okay, actually, the by pour down by IP, so they can find this 10 and this gene are complex together. Okay, so another way to analyze this data is to find the genotype and phenotype relations, right? As I mentioned previously, they can do the clustering at this dimension as 2 dimensions, either the gene expression and also the perturbations, right? They find 36 perturbation clustering and 37 gene expression clustering. Okay, so with each gene perturbation, they can calculate the scores for each gene expression cluster, right? For example, the UPR, and ISR. UPR is unfolded protein response. So you can see cluster or the gene set or the pathway, etc. Doesn't matter. So if more genes are more genes in this gene set changes significantly, then it has a higher score. That means the knockout of these genes will cause activate this pathway or this gene set. Okay, so you can see this the activator of this UPR is mainly like this ER-resistant chaperone or translocation machinery. And while for the integrated stress response, it will affected by mitochondrial genes and amino acid tRNA synthetase, etc. Okay, these genes. For the myeloid differentiation and erythroid differentiation score. So this is for differentiation, right? And we can see they also have different genes that are cause different differentiation directions for the cells, and especially for the myeloid differentiation, they find like mediator, the MED12. This is a mediator component, and also the KDM gene and the PTPN. So they belong to different pathways, right? So the authors also test whether they have synergy effect, right? For example, so they knock down this PTPN1, it will cause some anti-CD11B. So this is a marker for the myeloid differentiation. And the knockout of KDM will cause some difference, will cause the phenotype, while knockdown both will cause a much stronger effect compared to knockdown the individual ones. That means they have synergistic effect. So that means for the differentiation, they will have different pathways that are common. They will commonly they will work together to change the cell differential direction. Okay, and also composite phenotype, right? As I mentioned previously, they have the transposon element, the total mRNA and total RNA. So they can find that the exosome genes are quite related to the TE expression, while the mediators are related to the RNA. So this may be related to the splicing. Okay. And also they explore the genetic drivers for the chromatin stability. Okay, the chromatin stability, they use the single cell Lamb score. So this score is different from the scores that measure the single axis for single axis changes. But the overall changes of all the genes, the higher single cell average score means a higher global change of the genes. That means this may relate to the chromatin instability. So this figure shows that the differential expressed gene numbers are positively related to the Lamb score. That means so this is reasonable, right? If the global change, if the knockout of the certain gene causes a global change, or the chromatin instability, that will cause higher number of differentially expressed genes. Okay, so you can find this these genes that the CMPC, right? These genes are related to the chromatin instability. So with a TDK, so they measure the TDK knockdown cells compared to non-target cells. They can find a global changes of gene expression within each within different chromosomes. Okay, I think the author also mentioned about the mitochondrial genome gene expression changes, and I will not speak too much about the details of that part. And I think these examples have showing the power of this kind of single-cell CRISPR screen at genome-wide scale, and maybe there are a lot of things worth digging. I think this data maybe can be further analyzed. Maybe it can lead to a lot of other discoveries, but I think these are some kind of basic analysis showing in this paper. I hope you find this paper useful for your study, and we can think about together how to further analyze this kind of data. And I think in the future, this may be a regularly used technology to map the genotype and phenotype in different scenarios, like transcriptional or epigenetic regulation or transcriptional regulation between genes, transcription factors, epigenetic regulators, and the downstream gene expression change, right? In cancers or the stem cell or the cell development in diseases, etc. Okay. Okay, I think that's the kind of end of this video. If you find this helpful, don't forget to like the video and smash the subscribe button for my upcoming updates. Okay. Thank you very much. I will see you next time. Bye bye.